Well, I want to go through, I think the first thing is in terms of, we've been talking about the different size chips and the panels we've been looking at, so I want to talk a little bit about the different sizes we've been taking a look at. And some of it's pretty amazing in terms of the amount of data that we're collecting relative to what working with just a few years ago. Make a distinction between information and data. It's very clear that the larger chips generate more data. That's not really our goal. As a statistician, run into a lot of scientists and researchers coming in and figuring if they've got a lot of data, they've got a lot of information, and it's not the same thing. Whoops. Then I'm going to take a look at a couple of studies. One done here at Mark and another one done at Iowa State, comparing the evaluation of MBVs built with a 50K and an HD panel, and a little bit in terms of where we go from here. And I don't know, I take a look at these numbers and they just look like numbers after a certain while. So I kind of wanted to think about how I could kind of visualize in terms of what the changes are over there. And so I have my 128K SNP panel there. So I'm going to show you several things in terms of the relative size. So the sizes in terms of it. And thinking back in terms of where we were a few years ago, I think the first time I was here, we were dealing with MBVs built from a panel roughly this size. Maybe I need to make it twice as big. Saw some things in terms of 3K panels. Okay, those are a little bit bigger. A lot of the stuff we're doing now are with 50K SNP panels, building in terms of there. So that little thing's looking a lot littler than that. This is the relative size in terms of each of those pixels you kind of think of as one SNP versus the 128 where we were a few years ago in terms of looking at this, and where we are now, I think a lot in terms of the 50K, and where we're kind of taking a look at now with the high density panels of 770. But I want to think a little bit farther into the future, and it's probably not that far in there. So this is the size in terms of sequence data in there, and I have a size the same as the 770. If I kind of go, whoops, back and scale everything else back, the other ones start looking a lot smaller. So still, if we're talking about big, bigger, biggest, we got a ways to go yet from even a 770 panel. But if we can, the one thing that got left off was the 128. That was kind of a little, the box around it was bigger than the the part that was colored in size in there. So we're getting a lot more data, and we will be getting a lot more data. And what we're seeing with that increased size of these panels is a dramatic increase in the amount of data per animal. If we kind of go through and we take a look between a 50K and a 770, that's roughly a 15-fold increase in terms of the amount of data we're getting. There's an increased cost per animal. Okay, you go from a 50 to a 770, twice as much. Likely to drop, but my guess is the 770 high-density panels will be more expensive than the 50. If they're not, then they're probably going to stop selling the 50. But even though there's an increased cost per animal, there's a decreased cost per SNP genotype that we get. We get 15 times the data twice. That's a 7-fold, roughly, decrease in the cost that we're paying for each of those SNP genotypes in there. And so if we're talking about spending the same amount of dollars, the question in terms of do we go and use 50K, do we use the high density, it's kind of a trade-off. If we use the 770, we're doing the same, half as many animals genotyped. Okay? So, but what we'll do is get roughly 7.5 times more SNPs genotyped in there. And so it's a question in terms of this trade-off here. Am I getting twice as much information for twice the cost? Ideally, I get more. Okay? Now, what we're interested in, and I was talking about, was it's information. We get a lot more data in there. And what we're trying to do, when we really get down to it, is predict future progeny performance. I mean, that's what we're looking at. Everything else is just kind of the window dressing. Okay? As I said, that high density panel is generating 15 times more data than that 50K panel. I feel very safe in saying it doesn't have 15 times more information. If it did, it'd be a no-brainer. We just kind of go through and use that. And you can see part of it. You saw a number of talks talking about imputation, imputating up from a lower density panel up to a higher density panel. And we can go through and with the 50K, get up to pretty close to 770 by filling in a lot of the data. Now I make that sound real easy. We talked about imputation, and I haven't imputed anything in there, so somebody else is doing the imputation. It's always much easier than if I'm the one having to do it. But to do that imputation, we have to have adequate supporting information in terms of either animals back in the pedigree, a large enough reference in there. And so what I'm really interested, does an HD panel provide sufficient additional information to kind of justify its cost? And I guess I could stop here and say, well, it depends. Okay? But I also wanted to go back and say, what would I think an ideal panel would look like? Well, I'd almost see that as being relatively small, and depending on what you're talking about, a small panel would be. And what I'd like to, especially if I'm going to be estimating MBVs from a panel, what I'd like it to do is to go through and contain just the set of all economically relevant functional SNPs. I'll have all of them on there, nothing else, and I could do probably a really good job calculating MBVs from that. As has been pointed out earlier, we don't know which SNPs those are. Got people working to try and narrow down into it, but it'd be really nice to have those. And even if we had them, we didn't know what the effect of those SNPs are. But my guess, if we could figure out what they are, getting the effects would be a whole lot easier if we didn't have a lot of extra stuff in there. I think in terms of whenever we're looking at people bringing data in and they measure 10,000 different things and they're saying, what does it all mean? If they come in there and they say they've designed this well and they've had, these are the things that we think are important, and I'm going through and modeling, I'm a lot more likely to be able to help them reach a useful conclusion as opposed to, well, we measured everything that possibly would be out there on 10 individuals in there. So if we can kind of get rid of some of that extraneous stuff. And so I want to kind of take a basic in terms of, does going to a larger panel generate a better MBV? And there's a lot of ways in terms of taking a look in terms of whether we want a 770, a high density panel or not. This is one aspect. We're just saying, well, if I have a set of 50Ks or I have a set of 770, will one of them generate a better panel? So these are a couple of studies that looked at building an MBV using both a 50K and a high density. We're going to compare them using the genetic correlation between the MBV and the trait. Genetic correlation is nice because it's bigger. We like seeing that. Spreads things out a bit more. If we're really interested in terms of kind of a real measure in terms of how well it's working, you need to be taking a look at the percentage of genetic variability that's accounted for the MBV, so basically a reliability. And I've got my formula up there, so I feel pretty good. You square the genetic correlation in there. And so as we're kind of going through, if I'm taking a look at a genetic correlation kind of in the range from 0.2 to 0.3 to 0.5, if I see something around 0.2, I'm accounting for just 5% of the variability. If I go and take that 0.2 and bring it up to 0.3, I'm getting up to 10%. And 0.5 gets me up to 25%. So I guess if you're looking at the SAIL literature, you'd rather put in these numbers than those numbers because they're bigger. Okay? And the first set of results are some work that Warren Snelling did and presented at BIF this year, and was looking at the genetic correlations for some cycle 7 trained MBV, trained using both a 50K and an HD panel for both birth weight and ribeye area. I have the genetic correlations here along with the standard errors there, and they look very similar to each other. Okay, Dorian Garrett did some similar work with dairy cattle. I think these were also presented at the BIF meetings this year. This was taking a look at a mixed breed trained MBV, and then he's going to take a look at evaluating 3 different breeds, two different breeds in the cross and Holstein for milk, fat, and protein. So these are correlations and taking a look at their performance in the next generation. 50K, 0.71, HD. Again, we see very similar results in there. Same MBVs, now taking a look at it within a different breed for Jersey. I get very similar results in terms of whether I'm using, well, Dorian got very similar results whether using a 50K or an HD. And in crosses, again, very similar results in there. Also, well, you say, well, maybe if we go up to 770, we'll get something that's a little bit more robust across breeds. So if I go through and I'm doing a single breed training, so Holstein, trained within Jersey, trained within the crosses, and I go through and along the diagonal, I have it evaluated in the breed it was trained in there. If I go and evaluate it in a different breed or different cross from training, we get the result that we pretty much expect, that we see a drop in terms of its ability to predict. Actually, these don't look too bad. So these are 50K results. Here are the HD results. The within breed values are very similar. The go across breeds in there. So I'm not saying in terms of there being a whole lot more robustness in terms of what Dorian was finding. Okay. So if we take a look in terms of moving from a 50K panel to an HD panel, HD generates a lot more data. Okay? And if we take a look at that, it isn't sufficient to generating that more data. It doesn't guarantee that we're going to generate a lot more information. If we just kind of go through and say, well, I'll go through, use a higher density chip, and say the implication is going to be I'm automatically going to go through and get a better MBV. That doesn't necessarily follow in there. And so if we're taking a look at improving those genetic predictors based on genomic information, that's going to require increasing the amount of information. Other speakers have talked about that. And kind of like thinking about the 3 main types of information going in there, a big part of that is having information on phenotyped animals, collecting phenotypic information. We do a lot better job picking the low-hanging fruit. Okay? Traits for which we've been collecting phenotypic information over a long time and have a good database of, that's going to be the key in terms of going forward in there. Along with that is having a broad spectrum in terms of genotyped animals. You can't just go through and genotype 10, okay, in there. And then it's going to be the number of SNP that you have genotype for. I kind of remember talking about having these MBVs trained using 156 quality trained SNP a while back, and the genetic correlations I was putting up there, I recall, were kind of disappointing. So is bigger better? Well, going from 128 to 50K, the answer would have been yes in there. Okay? And I think it's a question in terms of balancing these three things out. And as we go forward, as we go and start increasing the number of genotyped animals, that's going to increase our ability to take advantage of having greater number of SNPs. So as we go forward, what we see today isn't going to be the same answer if I come back here in 10 years. Probably not going to be the answer next year. Seems like these change very quickly. Okay? And so part of it is, where do we go from here? And I borrowed quite a bit this from Warren's talk that he gave at BIF. What we really want is to end up with a genetic predictor based on economically relevant functional SNPs. I think that would be the ideal. I'm confident when I come here next year we won't be there. But that's kind of where we're heading. There's a lot of tools coming into that. You've seen some talks in terms of that getting DNA sequence information is coming along. Okay, so we're starting to get more and more of that. So that's going to give us a lot more depth in there. We also can collect data in terms of gene expression and regulation. That's going to start telling us in terms of which genes are active. Okay, tell us in terms of how things are being regulated. We're going to need to start tying these things in terms of gene networks. Okay, they don't operate in isolation. We usually think about, I've just got these 50,000 independent things, but they interact in terms of networks, and these might help us identify in terms of where the keys are. But we also need to think about moving up the scale in terms of starting to bring in things at the systems biology level. So it's going to need to be bringing things in together at a lot of different scales. It's going to require a lot of very creative people. We need to couple that with having an adequate resource and building a resource in terms of genotyped animals. That's coming along. But I don't want to lose the fact that this all stands or falls in terms of having that phenotypic information. There's not going to come a time where we're going to be able to toss aside and say, I don't need to collect phenotypes anymore. I'll just have to get genotypes. Okay? At least not in my lifetime, I don't think. Okay? And when we take a look at this and we're interested to see whether this is worthwhile or not, we've got a lot of stuff happening here at a very low scale, at the molecular scale. It's very exciting. It's giving us lots of opportunities, a lot of promise there. But the worth of these advances at the molecular scale, okay, will ultimately be measured at the production level. If it doesn't pay out the production level, we're entertaining ourselves in there. This is where we need to be, and that's going to depend in terms of kind of going back in terms of having those phenotypes. Thank you. Any questions?